Hello guys, welcome back to part 2 of DNA Code of Life. So we're going to talk about how a protein is made now. Let's talk about the process of protein synthesis. It has two parts. The first part is transcription. And this whole process of protein synthesis, it takes place in a cell, but transcription takes place in the nucleus. And they will ask you to describe the process of transcription. So what happens is that the first point, the double helix, which is the DNA molecule, it unwinds. So it will move from this shape to this second shape here. And then the double-stranded DNA, it will unzip, and then therefore the weak hydrogen bonds will break. When it unzips, it moves from this shape now to this shape. To form two separate strands, we have the two strands there, and then one strand is used as a template and is going to form mRNA. Using free RNA nucleotides from the nucleoplasm, and then the mRNA is complementary to the DNA. mRNA now has a coded message for protein synthesis. Now let me talk about the nucleotide, then the nitrogenous bases part. So one strand is going to form mRNAs, but then how does it form mRNA? It comes with nitrogenous bases specifically for whatever that needs to be repaired. For an example, let's say that the cells of your hair needs to grow, and then the protein that is made here is specifically for that. The DNA will come with the specific nitrogenous bases. For an example, let's say it comes with A, G, T, and C, G, C. So A, G, T, and C, G, C, those are called base triplets. They are called codons in mRNA. So what's going to happen? The mRNA that is going to be made will be made with the nitrogenous bases pairing with those nitrogenous bases on the DNA. So how do we pair them? Remember I said A pairs with thymine. But then you cannot write thymine in mRNA. So instead of thymine, you're going to write U for uracil. So A pairs with uracil. And then we have guanine there. Guanine always pairs with cytosine. You're going to write C there at mRNA. Then T there. For T, we're going to write adenine. It's going to pair with adenine. C pairs with guanine. G pairs with cytosine. C pairs with guanine. So the mRNA that is going to be formed, it's going to have these nitrogenous bases. They need to pair with the ones from the DNA strand. So now after it is made this messenger RNA, the base triplets, the base triplets, which is UCA, the first base triplet, GCG, the second base triplet, they are called codons. And then the mRNA is going to leave the nucleus through the nuclear pores, and then into the cytoplasm. That's where translation starts. So this process of translation, it happens in the cytoplasm. So what happens is that each tRNA, now we have an RNA that is called a tRNA, T for transfer RNA. They come with amino acids. This organelle here is ribosome. The tRNA on the ribosome there, we have two of them. As you can see, they are carrying amino acids. Amino acids are those round circle things that I just drew there. So they are also going to pair. They're going to have the nitrogenous bases that correspond with the ones on mRNA. So how are we going to pair them? The first base triplet is UCA. It's going to pair with the first tRNA there. U pairs with what? With A. C pairs with guanine. A pairs with U. Now the second base triplet, GCG. G pairs with C. C pairs with G, and then G pairs with C. That's why on tRNA we have AGU, and we also have CGC. They must correspond with the ones there on mRNA. And remember, the ones there on mRNA correspond with the ones on DNA that we talked about. So how are you going to write it if in the answer sheet, if they ask you to describe this process? You're going to say each tRNA carries an amino acid. When the anticodon on the tRNA, anticodon is the base triplets on the tRNA there. When the anticodon on the tRNA matches the codons on the mRNA, they need to match or correspond. The tRNA brings the required amino acid to the ribosome. Those circles there that I drew on the tRNA, those are amino acids. And then amino acids, they will become attached to each other. Look there under the cell, I drew 2 circles, and then they have a line in the middle, which is a peptide bond. Amino acid become attached by a peptide bond, and then the amino acids combined together are what we call a protein. The bond is a peptide bond, and then our protein is now made. So this is all about protein synthesis. Let's look at the biological terms to see if we talked about all of them. The peptide bond, we talked about it, is that bond between amino acids, right? Uracil is the nitrogenous base that is only in RNA. Thymine is a nitrogenous base only in RNA. Ribosome is that organelle whereby the tRNA brings the amino acid. And then we have ribose sugar, that's the sugar in RNA, deoxyribose sugar in DNA, hydrogen bond, those bonds that are between nitrogenous bases in DNA. It's a weak hydrogen bond. Translation is the second process of protein synthesis. Transcription is the first one that happens in the nucleus. Double helix is the natural shape of a DNA. They will ask you this in your biological terms. The nucleotide, we talked about it. It's the one that has phosphate, sugar, and nitrogenous base. And then we have a template. A template, we didn't talk about it. We have messenger RNA. That is the RNA that is made by the one strand of the one that is used as a template. The strand that is used as a template is that strand that forms the messenger RNA. Transfer RNA is the tRNA we talked about. Protein synthesis is the whole process. Codons, we talked about codons. They are the base triplets on the messenger RNA. Anticodons, the base triplets on the tRNA. So this is how the process is going to be drawn to you. They might draw it this way and ask you questions, and I will answer